Today's topic, SC Framework, the Dimension Reduction and Clustering Notebook. If you haven't watched the previous videos, we recommend you to go back and watch them first. In this notebook, the aim is to group cells based on how similar their gene expression profiles are. Ideally, the resulting clusters should represent different cell types. This grouping is achieved in two steps. First, the embedding and then the clustering. Embeddings are dimension reduction methods to transform high-dimensional data into lower-dimensional representations while preserving the inherent structure and relationships between individual cells. The SC toolbox supports the Uniform Manifold Approximation and Projection, or UMAP, and the T-Distributed Stochastic Neighbor Embedding, or t-SNE, as methods for dimension reduction, with UMAP being set as the default value. Single-cell clustering is used to group individual cells into clusters based on similarities in their gene expression. The clustering allows to identify distinct cell types and characterize cellular heterogeneity within a population. The SC toolbox supports the Leiden and the Louvain clustering methods, with the Leiden clustering algorithm being newer and recommended to use. Visit our documentation to learn more about the differences between those methods. Coming now to the notebook, we start by loading all required packages and the configuration file. We will continue the analysis by loading the batch-corrected data that we saved in Notebook 3. Then, we have to set the initial parameters, starting with the condition_column parameter, which should contain the condition that we want to investigate. In our example, we are comparing the different time points, so we will set condition_column to time point. The threads parameter lets you set the number of threads used for multiprocessing. Next, we have to choose the embedding algorithm used. We will stick to UMAP. Since our clustering depends highly on the output of the embedding method, choosing optimal parameters for UMAP or t-SNE is important. But finding the optimal parameters is not a trivial task, and there is not one default parameter that works for all datasets. To overcome this, the notebook contains the search_parameters option. If this is set to true, the notebook calculates and shows multiple embeddings using a given range of parameters. This lets you decide on the optimal parameters for your dataset. The different ranges can be set here. For UMAP, this is the min_dist and spread parameter. The min_dist parameter sets the distances between points to make the plot look more clustered. On this example, you can see how a 3D visualization of a mammoth is represented in 2D with different min_dist parameters. The spread parameter sets the effective scale of embedded points relative to min_dist. For the UMAP, there is one more parameter, the n_components parameter, which lets you choose into how many dimensions the data should be reduced to. In general, we recommend setting this to 2, but you could also set this to 3 to get a 3-dimensional UMAP. For t-SNE, there is the perplexity parameter, which is related to the number of nearest neighbors accounted for during embedding creation, where larger datasets require bigger values, and the learning_rate, which sets the learning rate important for embedding cluster formation. The learning rate should be set in the range of 10 to 1000. The ranges are each given in a tuple with 3 values: the start, end, and step value. While the start value is included in the range, the end value is not included. Here you can see one example with start set to 0, end set to 5, and the step size set to 1. The resulting range is 0, 1, 2, 3, and 4. The clustering parameters are similar to the embedding parameters. We first have to set the search_clustering_parameters option. If this is true, multiple clusterings are done using a range of resolutions. The resolution parameter determines how many clusters we get. The higher the resolution, the finer the clustering becomes, resulting in more clusters. A fine balance is needed to avoid both overclustering, which results in splitting biologically meaningful groups, and underclustering, which results in combining biologically distinct groups. The resolution parameter is identical for both clustering methods. You can either use Leiden or Louvain. We will choose Leiden since it's faster and more robust. The initial resolution should be chosen based on how well the clusters fit to the displayed shapes. However, the biological background should also be accounted for. For example, if we expect rare cell types with low number of cells, we might want to choose a higher resolution. The last parameter, cluster_columns, lets you choose the number of plots shown in each row of the clustering grid search figure. After setting all parameters, we continue with the embedding. The resulting plot shows a grid of multiple embeddings with different parameter combinations. Since we selected UMAP, the different min_st parameters are shown over the columns, while the spread parameters are shown over the rows. After visually inspecting the results, adjust the parameters in the input field for the best embedding. While it is somewhat subjective what the best parameters for an embedding should be, the chosen embedding should display clear structures that are neither spread too thin nor too clumped up. In this case, we chose min_st equals 0.1 and spread equals 1.0. We can ignore perplexity and learning_rate in the input field, since we are not using t-SNE. The last cell in this section then calculates the embedding with the final parameters. Next, we look at the embedding colored by a variety of quality measures. The visual inspection is a critical step in data analysis. It helps us understand data structure and validate whether our parameters produce expected results and assess quality metrics to ensure they do not dominate the embedding structure, since we want the gene expression to be the primary driver. The first plot overlays the quality metrics from Notebook 2 onto the UMAP. This helps to identify if quality measures disproportionately influence the embedding. In such a case, we recommend to go back to either QC or normalization and batch correction to adjust parameters in respect to the offending metrics. The second plot shows how the condition, in our case the time points, are distributed across the UMAP. The third plot highlights cell concentration per cell cycle phase, as well as per condition. Overall, we can see that some time points cluster separately, which is expected in early development due to significant gene expression changes across stages. With the embedding done, we can now assign each cell into a cluster. Similar to the embedding step, a grid search helps us to identify the optimal clustering resolution. The best choice depends on the structural clarity, so if the resolution captures meaningful patterns in the embedding. Also, knowing how many different cell types you expect in the data can also help, since clusters should ideally represent distinct cell types or subgroups linked to biological annotations. After visual inspection, we select resolution 0.1 for further analysis. This rather coarse clustering captures major cell types, but also allows for subclustering of individual groups later on. All clusterings with the different resolutions are saved in the anndata.obs table. The selected clustering can additionally be revised. You can use the recluster function to iteratively adjust clustering. The two cells are provided as a template for either joining or splitting clusters. Fill in the list at the top of the respective cell to activate joining or splitting. You can copy the cells as needed for more combining and splitting steps. For now, we do not want to adjust the clustering, so we skip this step. The final clustering will be saved in a new column in anndata.obs called clustering. After revising and adjusting, the final embedding is plotted colored with the condition and the final clustering. Next, we plot the number of cells in each cluster grouped by the condition. Clusters with very few cells may be statistically insignificant and should be considered for removal. Also, this plot lets you identify how well distributed the different conditions are over the clusters. Depending on the research question and the given condition, the individual clusters should either be homogeneous or evenly distributed across all clusters. In our case, earlier time points mostly cluster together, and also later time points mostly cluster together. This suggests temporal patterns in gene expression that align with developmental stages. If you are using UMAP and have set the number of components to 3, the next section allows you to do a 3D UMAP plot. However, this is disabled by default, since the process of optimizing in the 3D or higher dimensional space usually degrades lower dimensional representation of the UMAP. We skip this step since we set the number of components to 2. Lastly, we save the clustered in data into an H5AD file. With this step, we have completed the clustering notebook. If you plan to use this or one of our other notebooks, please check out our Docker image, which comes as a full functional environment for the SC framework to be used within a web browser. The next video will focus on marker gene computation. If you need further assistance or want more information, be sure to check out our documentation or visit our GitHub repository. Thanks for watching.